Hello, everyone, and welcome. We're so glad you could join us for today's webinar on clinical translation and cell and gene therapy. My name is Ellie Arnold, and I'm the training specialist at Stem Cell Network. We're excited to be co-hosting this session with our longstanding partner, BioCanRx. Today's webinar is designed especially for trainees and researchers who are curious about what it really takes to move discoveries in cell and gene therapy from the lab bench to the clinic. Our goal is to give you a practical, accessible introduction to the principles of clinical translation and hopefully make this complex process feel a little easier to understand and navigate. Before we begin, I want to take a moment for a land acknowledgement. As we're gathering virtually from locations across the country, we recognize and honor the Indigenous peoples of all the lands on which we work and meet. At SCN, our headquarters are on the traditional, ancestral, and unceded territory of the Anishinaabe Algonquin Nation, and we pay respect to all Indigenous nations across Canada. For those of you who may be new to Stem Cell Network, we are a Canadian not-for-profit organization funded by the federal government to advance regenerative medicine. We support research, training, commercialization, and knowledge mobilization, creating opportunities for our community to excel, and ultimately to help regenerative medicine therapies reach patients. I'm particularly excited about this webinar because clinical translation sits right at the heart of Stem Cell Network's vision, transforming lives through regenerative medicine. That transformation doesn't happen by accident. It depends on a skilled, collaborative community that understands not just the science, but also the regulatory, clinical, and commercialization pathways involved. My hope is that today's session gives you insight into the complexities of translation and maybe even spark some ideas about where you might fit into this evolving ecosystem. With that, I'd like to welcome Julie Jonkins, who will share a few opening remarks on behalf of BioCanRx.
Thanks, Ellie, and welcome everyone. As Ellie mentioned, my name is Julie Jonkins, and I'm the Training and Research Manager at BioCanRx. For those of you who may not know BioCanRx, we're a federally funded national network of researchers working to accelerate the development of leading-edge immuno-oncology therapies for the benefit of Canadian patients. And we invest in translating world-class technologies from the research lab into clinical trials. We also train and develop the talent needed for a thriving Canadian health biotechnology sector. And as Ellie mentioned, we're really pleased to be co-hosting this webinar with Stem Cell Network. We really often hear from trainees that while they may get exposure to developing new cell and gene therapies in their research projects, they're not often exposed to what comes next. So we've put together this webinar to hopefully help fill those gaps and help you better contribute to your research and also prepare for careers in translational research. A few housekeeping notes. We will record the webinar and we'll be posting this to YouTube afterwards so you can rewatch later. And also, we've dedicated some time at the end of the webinar for the Q&A, so please hold your questions until the end. And with that, we thought it would be fitting to have a trainee as our moderator for today's session. So I'm very pleased to introduce Matthew Jeffers. He's currently pursuing a Ph.D. in epidemiology at the School of Epidemiology and Public Health at the University of Ottawa with Doctors Manoj Laloo and Dean Ferguson. Matthew's thesis research seeks to address optimization of preclinical experimental design and analysis to maximize validity, as well as methods of assessing preclinical evidence to better inform decisions to translate novel cell therapies to early phase clinical trials. Over to you, Matthew.
Thank you, Julie, and thanks, Ellie, for the introduction. And thanks to Stem Cell Network and BioCanRx for putting together the lineup of speakers that we're going to have today. I'm pleased to present our first speaker, Dr. Rasini Wiratna. Dr. Wiratna is the Director of Disruptive Technology Solutions for Cell and Gene Therapy program at the Human Health Therapeutics Research Center of the National Research Council of Canada. In this role, she provides scientific leadership and strategic vision for the program, which is aimed at developing innovative technologies to help enable affordable engineered cell and gene therapies for chronic diseases and rare genetic disorders affecting Canadians, as well as coordinating a national effort to increase accessibility of these therapies. Prior to joining the NRC, Dr. Wiratna was a senior principal scientist at Pfizer Vaccine Research and associate director of pharmacology at Coley Pharmaceutical Group. Dr. Wiratna is a Fulbright and Killam Scholar with over 25 years of experience in the biopharmaceutical industry, holding a MSc in bacteriology from University of Wisconsin-Madison and a Ph.D. in microbiology and immunology from Dalhousie University. So whenever you're ready, feel free to share
Thank
your
you very
slides.
much, Matthew. I will share my presentation now.
Yeah,
Yes.
it
Yep.
looks
All
all
right.
good.
So thank you very much. And it's my great pleasure to open up this webinar. So in my talk, I will walk through how cell and gene therapies move from discovery in the lab to clinical translation, highlighting the transition points and challenges that make these therapies different from traditional or conventional drugs. I will focus my part of the talk mainly on the preclinical development phase of this clinical translation and clinical development of these life-saving therapies. But in later talks, you will hear about the regulatory pathways for moving these therapies from bench to clinical trials into approval, commercialization, and broader access. And you will also hear case studies of Canadian therapies that have transition from the bench to clinical trials. So as you all know, in the last decade, we have seen major leaps forward in the field of cell and gene therapies. Gene editing, using all these novel technologies listed here, allows precision DNA correction, improved delivery systems, both viral and non-viral, offers targeted delivery of these genetic material into tissues or cells of interest, increasing both their efficacy as well as safety. Engineered cell therapies, such as CAR-T therapies, have shown curative potential in cancer and now also in autoimmune diseases and other chronic diseases. And stem cell therapies are revolutionizing the regenerative medicine approaches. And alongside with all these biological advances, manufacturing technologies, including automation and closed systems, have really made clinical grade products feasible, in particular when you're trying to clinically de-risk this through clinical trials. So these advances collectively make cell and gene therapies one of the most disruptive areas in human medicine. So what is really clinical translation? Clinical translation is the process of turning scientific discoveries into therapeutic applications that benefit patients. When it comes to cell and gene therapies, this means translating complex biological innovations like engineered cell therapies and gene therapy tools into safe, effective products. So as I mentioned, I'm going to really concentrate my talk on the preclinical phase of things. So when you talk about preclinical development of these therapies, this preclinical phase can largely or broadly be divided into two phases. The early discovery proof of concept phase, which focuses on identifying the therapeutic target and demonstrating biological efficacy in relevant models, followed by the next phase that focuses really on translational and regulatory readiness. So generally, this very first phase of preclinical development really sits outside direct regulatory oversight. As long as the products that you're making to test in this phase are really restricted to your preclinical work, whether it's in vitro laboratory work or animal studies, and these products are not intended to be used in humans. Even though it doesn't have direct regulatory oversight, the developers are expected to follow sound scientific and ethical research practices, even through this very first phase. So the very first phase of the real outcome of this very first phase of preclinical development is to come up with a well-characterized candidate with demonstrated potential to treat the targeted disease that is ready for more formal preclinical evaluation in the next stage of preclinical development. So at the lead confirmation or the clinical trial enabling study stage, before you transition into the second stage, the developers are really encouraged to seek consultation with regulators. And this really helps you to identify key data that the regulators are expecting to see in the clinical trial, in a clinical trial application, because the second stage is really focused on translational and regulatory readiness, generating data that is necessary to move into human clinical trials. So the data that is generated in the second stage, this really forms the key data set that is going to support regulatory filings to help you move your therapeutic from preclinical stage into clinical research. So when you move to the second stage of conducting clinical trial enabling studies, quality becomes a very, very key thing. You need to make sure that you're conducting the studies in this stage under good laboratory practices, or at least under the principles of good laboratory practices. And good laboratory practices are set of quality standards that ensure reliability, consistency, and integrity of non-clinical data that are supporting clinical trial applications. And in this stage, you will also be establishing manufacturing processes to manufacture your therapeutic product. And you need to make sure that you establish processes that are reproducible and well controlled and in compliance with good manufacturing processes. And with defined product specification, release criteria, and potency assays. So this really means that you need a multidisciplinary team to help you navigate through this preclinical stage. And this means, having, depending on your therapy that you're developing, molecular biologist, especially in this second stage, having toxicology consultants helping you to come up with the necessary studies to show safety and toxicity of your therapeutic, pharmacologist, process engineered, regulatory consultants, and in particular, quality specialists that ensure quality assurance and quality control of your data. And along with this cross-disciplinary team, you will also require infrastructure to do GMP compliant manufacturing, animal testing capabilities, and other things like that. So moving into the quality aspect of things, which is really important, particularly the second stage of preclinical development. So as I mentioned, the requirements for quality changes as you move from your early discovery into clinical trial enabling studies. So for that, you need to ensure that all non-clinical studies that you conduct in that second preclinical phase are planned, performed, monitored, recorded, reported, and archived in a way that guarantees scientific integrity and regulatory confidence. So what does that mean? That means having standard operating procedures that outline the protocols that you're using to conduct your experimentation. And the studies that you're doing, you write study protocols that are reviewed by your study directors and are reviewed and approved by your quality assurance team. And once your studies are completed, you should write formal study reports with the data that goes into those reports appropriately QC'd and looked over by the quality assurance team. And the experiments are conducted by qualified personnel. Not just that they're conducted by qualified personnel, but you maintain their training records to show that they have been trained and they understand and have the expertise to conduct these experimentations. And when you're doing these experiments, you should really have an extra set of eyes to oversee and cross-check. Make sure that your calculations are correct, you're adding the right reagent at the right time, you're dosing the right animal with the right drug, and that this oversight is also recorded and signed off. So it's signed off by the person who's conducting the experiment, as well as the person who is conducting the quality checks. You should have a sample management system implemented so that you have a proper chain of custody for all the drugs that you're or test articles that you're using in your experimentation, and you record all the information pertinent to the reagents that you're using, the lot number, the storage conditions, expiry dates, and things like that. And that the equipment that you're using are well maintained and calibrated. And oftentimes when you're conducting these experiments, that you do it in a blinded fashion so that the operator, as well as people who are involved in data analysis, are not biased by knowing exactly, what drug is used for which animal group, for example. So in order to ensure this blinding, you should have a system to randomize animals in animal experimentations and to anonymize your sample. So overall, the very key is to have a rigorous quality assurance and quality control system set up, as well as ensure that you, all throughout this process, that you document, what you do, as well as your raw data and your data analysis, and retain these data according to data retention policies. So quality is key. Documenting what you do is key. If it's not documented, essentially, it was never done. So that becomes a key component, especially in the second phase of this preclinical development phase. So I'm going to just take you through an example of how at NRC we have applied this process to practice in developing a novel engineered cell therapy or CAR-T therapies targeting CD22 to treat pediatric and adult patients with CD22 positive B-cell malignancies, which is currently in clinical trial. So this was just a very high-level overview of our design. The early sort of target discovery, design, and development phase. So as I mentioned, this was a CD22 targeted CAR-T therapy. The antigen binding domain was an antibody targeting human CD22, and this was a llama-derived single domain antibody. So we immunized llamas with human CD22 extracellular protein. And we generated phage libraries using immune llama B cells. These phage libraries were panned, and we were able to come up with 28 unique CD22 single domain antibody sequences that specifically bound human CD22. And then we were able to screen these 28 potential VHH CD22 binders and narrow it down through a successive rounds of screening to 14, then to 8, and then to 4 lead molecules. So since this was our very first CAR-T therapy that we developed, in order to carry out this process, it was necessary for us to establish a number of tools and technology platforms. One of the key tools that we established was this modular CAR plasmid that allowed us to very easily swap in and swap out different parts of the CAR molecule, whether it's the antigen binding domain, the hinge domain, the transmembrane domain, the co-stimulatory domain, or the signaling domain. So this modular plasmid allowed us to very quickly design and make a library of CD22 CAR-Ts with each one having one of these unique CD22 VHH as the antigen binding domain, and be able to screen this library within a few weeks. To do that, we also established high throughput screening platforms, and following that, platforms to really optimize this CAR, optimize for hinge length, etc. So just to let you know, these tools that we developed and our assay platforms are available to share with the research community. So if there's anybody who is working in developing CAR-Ts in the CAR-T field, and if you're interested in accessing any of these tools, please feel free to contact me directly or get my contact information through the organizers. So through this first phase, having identified 4 lead molecules and tested them through very rigorous in vitro and in vivo assays, we came up with a lead and moved on to the next stage of really establishing the clinical trial enabling data set. So this is really that second phase of preclinical development that I talked about. So for that, we came up with very robust plans to carry out pivotal preclinical anti-tumor efficacy studies to demonstrate the efficacy of our CAR construct, workflows to show the safety and specificity of our CAR construct. We developed reagents that could be used as release characteristics in the release of our CAR GMP patient-specific GMP CAR T product. So once we've put together all these plans as well as a methodology to produce the lentivirus that is needed to produce the CD22 CAR and CAR, actual processes for producing the CD22 CAR T itself. We put all that information together into a pre-CTA information package, and we met with Health Canada for a pre-CTA guidance meeting where we presented our proposal. How are we proposing to conduct the confirmatory preclinical efficacy studies, safety studies, manufacture the CAR, lentivirus, and manufacture the CAR itself, and release testing characteristics. And we got Health Canada's guidance to make sure that what we were proposing were in line with what their expectations are for a clinical trial application. And once that was done, we went on to carry out these studies as well as establish robust processes to manufacture the lentivirus that is needed to produce the CAR T, as well as CAR T itself using healthy human T cells. And all this information went into our clinical trial application, which was put forth to Health Canada for approval. Upon receiving this approval, we were able to initiate the clinical trial date 2024, which is now is ongoing across seven different clinical trial centers in Canada. So that's just an example of that, and we've done similarly also to establish AAV a product, AAV based gene therapy product as well. So just moving on very quickly, generally the ones in the clinic, the cell and gene therapies overall follow the same general phases as conventional therapies in the clinical development phase, but the focus may be a little bit different. For the phase 1, it's really focusing, as with all therapies, focusing on safety, dose finding, but also looking at signals of efficacy as well as immune responses. Phase 2 is expanding on the efficacy and also looking at the durability of the response and refining the dosing. Phase 3 is confirming your outcomes in larger population, and phase 4 is really post-marketer surveillance. You will hear more about this in the next talk. But just also, unlike conventional drugs, engineered cell and gene therapies often follow what is called an accelerated or conditional approval pathway, reaching the market with a smaller and earlier phase studies than the conventional therapeutics. However, this does require extensive post-market monitoring to ensure long-term efficacy of these therapies. So regulators such as Health Canada, FDA, EMA are actively updating frameworks to accommodate cell and gene therapies, balancing both innovation and patient safety. So cell and gene therapies face unique clinical challenges compared to other conventional therapies, just because of their nature. They're biologically complex molecules. They're living drugs or gene therapy products that can behave. That behavior can be unpredictable in vivo compared to what we are seeing in vitro or even in vivo in animal studies. Dosing may be just one time, often one-time treatment, making the optimization a little bit tricky. Many of these target rare diseases or genetically defined populations, and that really limits your patient cohorts. Clinical trial design could be very much different to other conventional therapies where you may not have randomized controls or placebo controls, since this could be unethical to do so. Essentially, your clinical trials, your phase 1 starts with your patient population. Manufacturing is also very complex, and often they are individualized product because of their autologous nature. And this autologous product means that each patient's cells are processed separately, requiring very strict chain of custody, a chain of identity controls. And also, because the starting material is patient cells themselves, that adds to variability. So it's somewhat difficult to have internal controls within your patient group in your clinical trial. Also, the manufacturing process that you establish at lab scale may not necessarily translate to clinical scale. So you may need to tweak the process, but each tweak that you do to your manufacturing process may also impact your product safety or potency. So which means if you do change your manufacturing process over time, that may require new validation to prove that your material is the same as what you started from the beginning. And also the regulatory frameworks are still evolving as these therapies really push the boundaries of therapeutics compared to the conventional therapeutics that have been gone through clinical trials decades before. So expectations on preclinical evidence not only may vary from jurisdiction to jurisdiction, that may also vary as time goes on. So when you're developing these therapies, it's very important that you stay on top of current regulatory guidelines. And also regulators may demand long-term follow-ups to monitor potentially delayed effects. And there are also the ethical and safety considerations as well. So just to summarize, the key takeaways are clinical translation of cell and gene therapies demand integrated expertise, science, engineering, and regulatory strategy working together. The journey from discovery to clinic is really not linear. It's iterative and it's collaborative. Unlike traditional drug development, where it may be a little bit more linear, going from discovery, preclinical, clinical, and regulatory approval, cell and gene therapies may involve continuous feedback loops between scientific, technical, and regulatory functions. But despite these challenges, these therapies really represent a transformative frontier. And I don't think I need to say that to this audience. And continued advances in manufacturing, analytical, and trial designs will really accelerate their safe and effective translation into patients. So thank you very much. And with this, I will hand over to the next presenter.
Yeah, thank you, Dr. Virana, for walking us through that transition from discovery to preclinical to clinical. For our next talk, we're going to have Dr. Erin Bassett speak to us specifically about the regulatory pathways. Dr. Bassett joined BioCanRx in 2024 as the Director of Regulatory Affairs and Policy. Erin has scientific research and policy experience in academic, industry, and not-for-profit environments. She's passionate about translational research, evidence-based policy, and benefits of collaboration across sectors to realize the full potential of Canadian-led therapeutic development. Prior to joining BioCanRx, Erin was an R&D program director at Tetra Biopharma, where she led a range of activities, including non-clinical safety studies, drug formulation, manufacturing development, and preparation of clinical trial application packages for Health Canada and the FDA. Before her time in industry, Erin was a research associate at the Canadian Council of Academies. She also worked as a research associate at the Ottawa Hospital Research Institute, where she investigated the mechanisms behind pediatric brain tumor initiation. Erin holds a Ph.D. in developmental neuroscience from McMaster University, received in 2010. So Yeah, welcome, Erin.
Okay. Can you see my
We can see the
slides?
slides.
Just need to put them in presenter view now.
Yeah.
Okay.
Yeah, perfect.
Okay, great. Okay, thanks very much. Thanks for the introduction and for inviting me to speak here today. So I'll be following up that great overview that you just heard with some considerations through a regulatory lens, and I think you'll hear me echoing many of the same points. So just to zoom out a bit, most countries have a national health product regulatory authority. That's going to be ensuring the safety and the efficacy and the quality of quite a spectrum of products. And this will include small molecule drugs and biologics. And I've marked some examples on here, but there are, of course, many more. And there's Health Canada, which will be the focus today, and also the FDA, which will come up too. So to start off, a high-level overview of Health Canada. So Health Canada has the legal authority to regulate drugs and other products through the Food and Drugs Act, and then all of the regulations that are associated with that act. And it's organized into branches, and each one will have various directorates under it, which then further branch off into different centers or divisions, et cetera. And the directorate that I'm highlighting today is the Biologic and Radiopharmaceutical Drugs Directorate, or the BRDD. So BRDD evaluates what we call pre-market submissions for biologics, including cell and gene therapies, which means that it handles review of both clinical trial applications and submissions for market authorization of these products, so authorizing them for sale in Canada. One more point to highlight is the different cell and gene therapy products that have been market authorized by Health Canada. The majority are CAR-T or AAV-based gene therapies. And I will note that market authorization is just one of the many steps that actually provides patients with access to these products. But I think that's probably another webinar topic that maybe we can cover another time. But going back to how you get cell and gene therapies to clinical trial in the first place, when it comes to regulatory considerations, you'll often hear phrases like risk management, risk mitigation, risk-benefit analysis, and this really underlies a lot of the decisions related to administering these products to humans. So for first-in-human trials, as you heard, not only does the regulator want to see compelling biology, but they also want to see that there's an acceptable risk to start the trial. This includes a really well-justified starting dose. And they want to see that you've got a reliable product, as you also heard, which is manufactured under GMP conditions that are appropriate for the phase of the study that you're doing. And they also want to see you can characterize your product, for example, test its sterility and test its identity. And that there's a responsible trial design. So you've got a trial not just with a sound protocol that has objectives that you can achieve, but with something that, of course, protects the participants that are involved. So I think it makes sense to take a moment here to just appreciate the regulatory complexities for biologics in comparison to small molecules. I've got an image on the right here that shows a monoclonal antibody compared to an aspirin molecule, and the antibody is 800 times heavier, and that's just an antibody. When we get into cell and gene therapies, of course, the complexity is much greater. So small molecules, they're typically low molecular weight, relatively simple structure, well-defined. They can be made the same way each time, and you can really easily characterize by analytical methods to make sure that you've got the same active ingredient between your batches. And when you make a manufacturing change, you can also test that. In contrast, biologics are larger, much more complex. They come from living organisms or cells. They might be made by maybe a complex or a new type of manufacturing process. And the key is that there's inherent variation between the batches. And this is especially true for therapies made from individual donor or patient cells. For example, as you heard, CAR-T made from the patient's own cells. The starting material will always be different each and every time the product is made. So it means it's not possible to really characterize it fully the same way you would a small molecule. And it's also more challenging to assess the product before versus after you make a manufacturing change. So all of these things have to be factored in when regulators assess cell and gene therapies. And for the most part, Health Canada regulates cell and gene therapies as drugs, meaning they're subject to the Food and Drug regulations, unless the product is a donor-derived cell therapy that undergoes minimal manipulation and meets criteria under a different set of regulations for transplantation. So I guess the question then is, how do you navigate these complexities from a regulatory point of view? So you're not expected to figure it all out by yourself. One way is to have ongoing interactions with the regulator during the life cycle of your product. So you've already seen this type of graphic, but what I've shown here above are instances where you can interact with the regulator through a formal meeting, which is shown in yellow. And then when you actually make a submission, I put in green. So these are Canadian terms, and the key one, which you already heard of, is a clinical trial application, or CTA. And you can request this pre-CTA meeting with Health Canada to get their feedback before you submit the full CTA. In some cases, you can even have more than one before a clinical trial. And before you submit a new drug submission, or an NDS, which is for market authorization, you can also have one, or sometimes more than one, pre-NDS meeting. And we'll go in a bit into the details more in a few slides of these type of interactions. But I also just wanted to reiterate that, you heard this earlier, there's not a sort of one-size-fits-all development pathway for cell and gene therapies. You may not do a typical phase 1, 2, 3 approach. There's regulatory pathways that might allow these products to reach market authorization faster, while then collecting confirmatory data over a longer period of time. And there's quite a lot of harmonization between regulators. So the documents that you prep to submit to one regulator can kind of have you well on your way to submitting to another. And finally, there are many, many guidance documents that come from regulators or other bodies, such as the International Council for Harmonization, or ICH. So these guidance documents can cover everything you can think of. I'm flashing a few examples up here: considerations for development of CAR-Ts, specific non-clinical considerations, or something more general like how you do a CTA. And one thing that I'll just note as well is that Health Canada will sometimes refer to FDA guidance. They're a much smaller regulator, and they don't always develop their own guidance for every single thing that the FDA issues guidance on. So sometimes they'll actually point you to FDA guidance documents on particular issues. So the regulations and guidance documents also cover the good practices that regulators expect will be followed during the product life cycle. And sometimes these are referred to as GXP. And as you heard in the first presentation, these practices are where the idea of quality systems really come into play. You saw a great example of this in the first talk. And to echo that, I think this represents a really big difference between the discovery type work versus the work that brings the product to and through the clinical trial phases. So you've already heard about good laboratory practice, or GLP, in the first presentation. So I won't go into too many details on that, but just to echo that it's not going to be applying to your more discovery or proof of concept type studies, but it generally does apply to the kind of definitive non-clinical studies that are really key to assessing the overall safety and the rationale for putting your product into humans. So for example, toxicology studies that are key to supporting your clinical starting dose. And I will point out that sort of trying to turn a typical academic lab into a fully GLP certified lab is usually not something that's possible. But regulators understand that cell and gene therapies don't always fit neatly into the kind of classic small molecule toxicology programs. And that's where some flexible or hybrid approaches can sometimes come into play. So for example, if parts of a study could be done in-house while still following the GLP principles that you heard about in the first talk, and then some of the samples could maybe be sent to a fully GLP compliant testing facility for analysis. And then of course we have good manufacturing practice, or GMP. So this is part of quality management. It's going to ensure things like the proper design, the proper oversight and monitoring, the proper control of your manufacturing processes and also your facilities. And then it's assuring that the product is safe, pure, potent. So it's concerned with both production and quality control. And the GMP principles must be followed when a drug is administered to humans. One thing to note is that Health Canada is working on a guidance document specifically for manufacturing of gene, cell, and tissue therapies. And they recognize that decisions are, again, the risk word coming up, they're risk-based, and there's some GMP flexibility with the stage of product development. So for example, around things like evolving specifications for testing your product. And there are added complexities for cell and gene therapies. The FDA has recently released some guidance documents around this. For example, one related to potency testing of these therapies, and another about manufacturing changes and comparability. That's a really critical issue. And it is understood that development it's iterative, but especially once you've started clinical studies. A concern is that the manufacturing changes might negatively impact the quality of the product. So regulators really want to see risk-based assessments of any changes and whatever comparability studies you need to evaluate the product pre and post change. And echoing what you had already just heard, in some cases you can use analytical methods for comparability, but they may not be fully enough, and you might have to do more clinical or non-clinical or clinical studies as well. The final good practice that I'll touch on is good clinical practice, or GCP, just really briefly. It's a set of principles that govern clinical trials. So first, that they remain ethical, and also that they follow standards that ensure participants are safe and results are reliable. And I've noted here some clinical trial considerations. For cell and gene therapies, this includes things around small population and high unmet need. So they do have a different benefit risk profile, in that most can lead to sort of extended or even permanent effects. So first in human trials, as I'm echoing from the first presenter, will likely be in a subpopulation of patients with the disease rather than with healthy volunteers. Single patient studies are another option that might be relevant. You can submit a CTA for an individual patient to Health Canada. They'll usually review it quite quickly. And this can be a way to not just help a patient in need, but also to get some clinical insight, even if it's just in one patient. And a typical phase 1 will be to assess safety, inform your doses, maybe get preliminary efficacy data. And you also heard things about logistics in the first talk, right? So it's possible that you might be assessing feasibility in your phase 1, especially if there are some technical or logistical issues around the manufacturing or handling of the product. And for cell and gene therapies, single-arm trials rather than randomized control trials have been more common so far to support market authorization in many cases. There can often be approaches to sort of externally control or contextualize the data. And this can be based on things like the well-known history of the disease or using published data as a comparator. And a surrogate endpoint, which is something that's reasonably likely to predict clinical benefit, can be important to get earlier information about the product's biologic effect, especially for diseases with a long drawn-out course. And innovative designs as well, such as adaptive trials or master protocols, can also be considered. And there are various guidance documents that speak to clinical trials for cell and gene therapies. Health Canada has one for CTAs for cell therapy products, to point out an example. So to end off, I just wanted to cover some of the actual process when you engage with the regulator. So first, pre-submission meetings. So pre-submission meetings is a chance for you to get free and non-binding advice from a regulator before a clinical trial or a market authorization submission. So it's not mandatory, but it can be very helpful. And you can discuss your questions on a range of topics, manufacturing, quality, non-clinical, clinical, regulatory questions. And I've laid out sort of the pre-CTA process here. It does allow the regulator to familiarize themselves with you and your product development program. And it may be that some of the Health Canada reviewers at your pre-CTA will also be the ones reviewing your CTA. And how it works is you file a meeting request. And then when you get your date, 30 days before the meeting, you'll have to submit a briefing document to Health Canada, including information about your product, its manufacturing, what the studies you've done, some planned studies maybe, and the questions that you have. Health Canada will provide you with some advanced feedback shortly before the meeting in writing. And then you attend the meeting, you present, discuss with them, and then you're responsible for providing meeting minutes 2 weeks after the meeting. And I guess I will just point out that it's not the regulator's job to tell you exactly what to do. It's up to you to come to them with your data, your plan, your questions, really good rationale for your approach, and ask them for feedback. And even then, there's some instances where there's not always going to be a crystal clear answer that they can give you, but they will guide you and tell you the key considerations. And then once you're ready to do a clinical trial application, the information you submit to Health Canada is organized into the Common Technical Document, or CTD. And this is a format that's used worldwide for submitting to regulators. It's structured into different modules, and kind of the information that you have for these modules will vary as your development program progresses. And the first module, technically, it's not part of the CTD because it's region specific. So it's going to have documents and forms that are specific to Canada. And it's where you put in your clinical trial protocol and a really important document called the Investigator's brochure. That has non-clinical and clinical data about the product. And then module 2 will have key manufacturing and testing information about your product. And then you might have more manufacturing and quality information in module 3. And it's going to be, your product is going to be held to increasingly stringent manufacturing standards as you go from early to late clinical trial stages. And there's a 30-day review period for a CTA. And you have to be on your toes during this because you'll have, in most cases, 2 days to respond to a query from Health Canada during this period. So this is my final slide. Don't worry, Matthew. So to end off, I just wanted to emphasize that cell and gene therapies really do push the regulatory envelope in a lot of ways. And part of what regulators do is consider how they can modernize and innovate and keep pace with things. So there's across regulators globally, there's a lot of pathways, designations, programs that cell and gene therapies might be eligible for, ultimately to bring them to patients faster. So I've got a few examples, and some of the more recent ones were actually designed with advanced therapies in mind. Some might come into play more at the market auth stage, like reviewing your market auth submission faster via priority review. Others can give you more opportunities to get support and engage with the regulator during your product development stages. Typically, these pathways are for things that are addressing an unmet need for a really serious disease. And I just highlighted a couple at the bottom there. One thing regulators are considering is how data from one product can be leveraged for another similar one, sometimes called platform technology. And there's a lot of considerations right now around point of care manufacturing. And the UK's regulator has developed the first world first regulations for point of care manufacturing. So I'll stop there and thank you very much. And I'm looking forward to the discussion at the end as well.
Yeah, thanks a lot, Dr. Bassett. Really clear overview of the regulatory pathway. So we've heard about translation from this sort of systems perspective, regulatory lens. Now we're going to switch over to some real world case studies. So our next speaker, Dr. Megan Levings, is going to share her experience about moving regulatory T-cell therapies from the bench to the clinic. Dr. Levings is a professor in the Department of Surgery and School of Biomedical Engineering at the University of British Columbia and BC Children's Hospital Research Institute. After obtaining her Ph.D. at UBC in 1998, she undertook postdoc training in Milan in the area of immune regulation. During this time, she conducted pioneering work to define what we now know as regulatory T cells, or Tregs. Dr. Levings was recruited back to UBC in 2003, joining the Department of Surgery as a Tier 2 Canada Research Chair in Transplantation. Her lab has continued to advance research in Tregs, most recently using gene editing to redirect cell specificity and enhance their function. Dr. Levings is currently president-elect of the Federation of Clinical Immunology Societies and has won numerous awards, including the 2022 UBC Faculty of Medicine Distinguished Researcher in Basic Sciences and the 2025 Canadian Society for Immunology Award. So thank you, Dr. Levings.
Okay, thanks so much, Matthew, for the introduction and the opportunity to present as part of the webinar. Thanks so much to the previous two speakers for really setting up my talk, where what we've been trying to do over the last 5 or so years is put everything that they just told you about into practice. And as you heard, my lab works on regulatory T cells. And so what we're trying to do is bring forward the first, as far as I know, a Treg therapy in Canada. So this is just kind of a broad overview of all the different things my lab works on. And then on the right-hand side, the evolution of the clinical application of regulatory T cells as a therapy for many different disease contexts, most traditionally being used in the context of transplantation to prevent rejection, now more and more in autoimmunity, as well as many types of diseases that we didn't traditionally think of as immune-mediated diseases, such as degenerative diseases and various places where we need tissue regeneration. What I'm going to focus on today is the work we've been doing on a new cell source of regulatory T cells, and in particular, thymus-derived Tregs. All of these clinical trials over here, the vast majority of them have been done with Tregs that come from the patient themselves. So just like you heard for the CAR T cell product, a highly personalized autologous product that has a lot of benefits, but also comes with a lot of manufacturing and complexity, which results in an increased cost. And so several years ago, together with my colleague, Laurie West from the University of Alberta, we thought about trying to create other sources of Tregs that would be more like an off-the-shelf therapy. So using off-the-shelf Tregs is not a brand new idea. Actually, some of the initial clinical trials were with third-party Tregs, and probably the most well-known one is this study from more than 10 years ago using allogeneic cord blood Tregs that were partially matched to the donor in the context of hematopoietic stem cell transplantation. And as you can see here, the point of these regulatory T cells is to prevent graft versus host disease. And in comparison to historical controls on traditional immunosuppression, the patients who got the regulatory T cells had a much lower incidence of GVHD, suggesting that allogeneic Tregs, although eventually they will be rejected, do have potential for therapeutic benefit in this context. And so my lab's working on two different sources of allogeneic Tregs. What I'm going to talk about today is from discarded human thymuses, and another one that I won't talk about, but is accelerating work in my lab, which is deriving them from pluripotent stem cells. So you may be wondering where we get human thymuses from. So it's quite interesting that children who have different types of congenital heart diseases need to have their heart corrected. And in very young babies, the thymus is actually quite a large tissue and it blocks surgical access to the heart. And so the surgeons have to remove a portion, or in some cases, all of the thymus. And normally that would be discarded tissue that goes just into the garbage. So Laurie West likes to call this the garbage to gold project. And the idea we had is that since the thymus is the place where T cells develop, it's where regulatory T cells develop, that this could be a very excellent source of cells for therapy. So I'll spare you about 10 years of basic science research that really checked all the boxes to show that, yes, that is correct, and bring you fast forward to the manufacturing process that we've developed and we're now basically at the final stages of getting ready for the clinical trial application. So this is a fairly simple schematic diagram, and actually the overall process is conceptually very simple. We get the discarded thymus from the operating room across the street. My lab is at a children's hospital. The first thing we have to do is isolate the regulatory T cells. Then we freeze them because we don't have a GMP facility yet in Vancouver, although that's another project I'm working on. And we have to fly them to Alberta, where we then use the ACTM manufacturing facility to then thaw the cells, put them into culture, expand them, put them through various manufacturing processes, and then freeze them so that we would have an off-the-shelf product. So what I want to take you through today are just some of the big learnings that we had in trying to get this conceptually simple workflow into reality. So focusing first on the isolation part of the story. So as I mentioned, that had to be done here at BC Children's Hospital, but we don't have a GMP facility. And so we had to figure out how we were going to do that in the context of the environment we had here. So fortunately, as one of the previous speakers briefly mentioned, there's, in addition to the cell therapy guidance, Health Canada also has the cells, tissues, organs guidance, which is for minimally manipulated products. And so we were able to leverage that regulatory guidance to do the first part of this isolation under the regulations that would normally govern a transplant of like a bone marrow or an organ transplant. And BC Children's Hospital had the FACT accredited cell therapy lab. And so we were able to work with them to set up this process. However, they'd never done anything like this before, so we also needed to generate a quality management system. You heard a lot about quality. I never thought that I'd be hiring a quality lead for the project, but we now have one, Jessica Wilson, who's been really fantastic. And we also needed to organize infrastructure. We needed to renovate their lab. We needed to purchase equipment. And also because we're not allowed to work in this clinical environment on our own, we also had to train the clinical personnel to be able to participate in this rather lengthy process. So here's just some photos to bring it to reality. That's what a thymus looks like, kind of a bit of an ugly organ. Here's a technician in my lab, Jessica, working with one of the clinical staff, Adrian, processing the thymus in their clinical lab. And I'm not expecting you to read this, but this is a snapshot of the quality management system that Jessica Wilson set up for us. And so some of the things you can see here, everything in green is done. The things that are in red and orange are still being worked on. But you can see there's all these different steps that need to be done: training, completing the training forms, completing SOPs, change control SOPs, all the documentation for storage, the SOPs around the facilities, the environmental monitoring. So it's like, as a basic scientist, it was really an eye-opener to, I mean, I think we all do this day to day in our basic science lab, but in a not rigorous, well-documented, well-thought-out process. So one of the big learnings actually from going through all of this is how I can make my own basic science lab better by trying to implement some portion of this quality management system into the day-to-day stuff that we do all the time. Okay, so the other big learning from the Treg isolation process is that we decided to use an isolation system that was not GMP grade. And so in particular, we were using non-GMP grade antibody cocktails and magnetic beads. And at first, this seemed like a really great idea, but this subsequently came up with like a huge, it's solvable, but it's a huge additional logistical consideration, is that because they're not GMP grade, it means that the companies have not or will not give you any assurance of the quality of the hybridomas that are making those antibodies. So they will not provide you any information on whether they might have adventitious agents like viruses and prions or other things that could cause a risk for the ultimate human being. Furthermore, the antibodies are bound to these magnetic beads that are not GMP, and so we had to figure out a way to try and detect if there were any residual magnetic beads through our process. And so if you have antibodies that you're using that are not GMP grade, just be aware that you will need to test every single batch for adventitious agents, and that costs about $70,000. So sometimes you might be making decisions about using non-GMP reagents because they're cheaper to begin with, but you just need to keep in mind that down the road, that may not actually work to your benefit because you have to do all this extra testing. And furthermore, we're trying to figure out if it's even feasible to validate a mass spec-based assay to detect the potential residual beads. And if it's not feasible, what kind of risk analysis we're going to do. So yeah, the learning there was really to super early in the project, try and source what you think might be your GMP reagents down the road, and then do all your preclinical studies as much as possible with the exact research use only equivalents of those ultimate GMP reagents, because that'll save you a lot of headaches downstream. So fortunately, we have worked out this whole process, and this is just a snapshot of what the cells look like. So when we get thymus from the hospital, this is the regulatory T cells here in the unpurified state. And then at the end of our process, we now have frozen Tregs that are now more than 90% pure, expressing CD25 and CD4 and not CD8. Okay, so then the next stage is actually expanding those cells. So this is the point where the cells fly to the GMP facility, where it is now out of our hands. But another thing we had to do, spent a lot of time, is transferring our technology to the technicians that are working in the GMP facility who've never worked with this kind of cell product before. And there was a lot of back and forth. So something else to take into account in terms of timelines is, if you are yourselves not going to be doing the manufacturing, how are you going to efficiently transfer the technology to a whole another group of researchers who may or may not be familiar with your process. The process is fairly simple. We stimulate with beads that expand the Tregs. We have to grow them and maintain them. And then eventually at day 14, we freeze them. This is what the cells look like. So they start off being a reasonably pure population of Tregs, and then over time, actually, the population gets more and more pure. And one of the big challenges actually with our product is actually the sheer number of cells. So unlike CAR T cells, which can be grown in a relatively small chamber and the manufacturing is very well worked out, for our product, we're actually generating billions and billions of Tregs. So a challenge we had and continue to have and continue to work on is how do you actually manage these really huge culture conditions where we're growing liters and liters of cells. In fact, as part of our process, we have a cutoff at 6 liters because the GMP facility with our current bag-based process just cannot handle more than 6 liters of material. So that's also something else to keep in mind, like what's your vision for the number of cells you're going to be making and what kind of engineering strategies and biomanufacturing strategies are you going to use that actually enable that cell number to be produced. So just the last few slides, I want to come back to the quality testing, which has been mentioned a few times, just to highlight the journey that we had to go through with our quality testing. So at the end of the day, we need to be able to show to Health Canada that the cells that we've frozen are indeed Tregs and have the characteristics that we want and that they're not contaminated with other cells. So there's this protein FOXP3 that's the gold standard marker of regulatory T cells. So that obviously has to be measured as part of our quality testing. But there's no sort of off-the-shelf kit you can buy that's GMP certified to run on your flow cytometry assay. So we had to set that up ourselves. And moreover, in our particular product, we know that FOXP3 analysis is necessary but not sufficient to characterize the Tregs. And so we also need to be working on developing some functional assays like intracellular cytokine production, as well as suppression assays. And these are very complicated assays that are have a lot of variability and are therefore quite hard to standardize. And there's a third assay, which is actually an epigenetics-based assay that measures the methylation of the region of the FOXP3 locus. Very commonly, we do it all the time in my basic science lab without thinking. But trying to use this now in a GMP context is obviously much more complex. There's a recently released GMP compliant kit available from Thermo Fisher, but there's no guidelines on what the pass-fail threshold is going to be. The kit was backordered for like 6 months, and we still need to validate the assay for ourselves. So that's an ongoing process. So just taking you through the analytical validation of the FOXP3 assay, this was something that as an immunology PI, we've been running flow cytometry in the lab for 23 years. I said, what? We have to prove that our antibodies are specific for CD3 and CD4, but indeed we did. So we have 2 different immune phenotyping assays. One is the identity and the other one is the purity. And for these very simple flow assays, we had to figure out the limit of detection, the linearity, the accuracy, the recovery, the precision, the specificity, and the selectivity. And so Sabina Iveson, who is also on the call and available at the end for the Q&A session, set up this process to validate this flow cytometry assay. And furthermore, some of the work had to be done actually in the GMP facility, which is over here at the ACTM. And I mentioned this is a collaboration with Laurie West. And so we were lucky to do a combination of work in Laurie West lab, which is very close next door to the GMP facility. So the 2 technicians could talk to each other. And we set up these 3 parts of the assay to test. So one was the accuracy, linearity, limit of detection. One was the precision testing, and one was the selectivity and the specificity. So I'm just going to take you through what those experiments actually looked like. So for the accuracy and linearity and limit of detection, we set up an experiment where we actually intentionally spiked in non-Tregs. We used CD8 positive T cells so that we could test how well this flow cytometry assay could flag that there were non-Tregs in our culture. And then we also needed to figure out how low we could go in terms of detecting this contaminating product. So here are the different ratios of contaminants that were added in, up to 70% contaminating cells. And fortunately, this worked out really well. So as you can see from this little graph or chart, the theoretical percent of contaminating cells, the actual in the experiment observed contaminating cells. So you can see that we actually had very good correlation between the theoretical and the observed. And thankfully, that told us that this assay is very linear. And furthermore, the limit of detection was determined to be acceptable at 30%. Okay, and then we also had to go into precision testing. And so there's 3 different types of precision in your assays. This is also useful just for your day-to-day basic bench science. How repeatable is your assay? So if you do the same thing, if you do technical repeats in the same experiment, how well do they agree? There's something called intermediate precision, which is if you do in the same lab on a different day, but you do the same experiment, but with different operators, how well does that agree? And then there's reproducibility, which is the agreement between sites. So if I do the assay here or someone does the assay in Alberta, how well do the data agree with each other? And so that's where we set up this precision testing study where we had repeats done in Laurie West's lab and 3 different repeats with different operators done at the GMP facility. And here's just some of the results here. And you can see that fortunately, we had very good correlation between the different results. And so we ended up having really excellent CVs and great precision as a result of this. And so the Treg product assay passed with high precision of all these different types of precision, fortunately. Okay, so just 2 more slides. So big picture takeaways from this process is that you really have to take into account that everything takes much longer. I mean, this is true in research in general, but I think even much more true in this translational research. There's so many sources of delay. The costs are at least 2 to 3 times what you first think they're going to be. GMP reagents are incredibly expensive, but they can also help save you money down the road. So you have to do like an upfront cost-benefit ratio. As we talked about, you need to develop this meticulous quality management system, which is a huge amount of work. So you need to get a quality specialist on your team as early as possible. And yeah, as has also been mentioned, it's also important to consider that in these early phase clinical trials, the CTAs are flexible and you don't necessarily need to check all the different GMP, GLP boxes, but you have to have a good risk rationale for if you're not going to do the gold standard experiment, why aren't you doing it? And what is the risk associated with maybe doing a non-traditional approach? This is something that we continue to battle with, is creating redundancy because reagent shortages will happen. So just to give you an example, one of our key reagents, rapamycin, there was suddenly a 6-month worldwide shortage and we couldn't make timelines for the manufacturing that we had planned to do. So we had to suddenly switch and seek another supplier, which was challenging, and then do those crossover experiments to prove that this other supplier as reagent was equivalent. So that's something to think about when you're choosing your reagents. Is there only one source? Is there only one company in the world that's making this reagent you're going to need? Maybe not a good idea. You should think about having a backup plan basically for every single thing that's part of your process. And all that flow cytometry validation that I just talked about, of course, costs a lot of money, which we had not budgeted for at all in our initial work to get this funded. So the other day I was on LinkedIn and I saw this little quote and I thought this just really captures it all. Program management is the factor that determines CTA success. It's not just a regulatory submission, it's the coordinated execution of the cell manufacturing, non-clinical, clinical, bioanalytical, and regulatory strategies under tight timelines and scientific uncertainty. And in the Canadian academic context, you basically really need to be doing it yourself. You can hire consultants for some parts of it, but of course they're very expensive. They don't work for you and they don't have the overall perspective. So you really need to have 1 or 2 people on your team that have eyes on everything, the whole thing from the basic science all the way to the clinical trial. and then just bring in key support in very defined areas. So here's our clinical trial. It's going to be called the Titan trial. It's a dose escalation trial to test the ability of allogeneic Tregs to prevent GVHD. I'm not going to go through all those details, but we're super excited to be putting the finishing touches on the CTA and submitting it in the next couple of months. So I'm going to end there and just really want to acknowledge Sabina Iveson, who's been the project manager on this whole project, and a huge team of people who've been working with us along the way. So I'll stop there.
Thank you so much, Dr. Levings. We might not have a lot of time for questions at the end, so I'm going to welcome the speakers to, during the next talk, click the Q&A button at the bottom, maybe type out answers to some of the questions that should be available, so that everyone will get a chance to see some of those. And we're going to extend the webinar by a couple of minutes just to make time to answer maybe a couple questions that remain. So for our last talk, we're going to hear from Dr. Somya Viswanathan, who is going to share another translational journey to the clinic. Dr. Viswanathan is an associate professor at the University of Toronto and a senior scientist at the Schroeder Arthritis Institute and the Krembil Research Institute at the University Health Network. Her expertise spans both academia and industry, including her work on the development of regenerative medicine products at Johnson & Johnson, and the manufacturing leadership at Celcan and OIRM. Her research is focused on developing cellular and immunotherapies that are anti-inflammatory and primarily focused in osteoarthritis. She's conducted several clinical trials, including using autologous bone marrow-derived MSCs in a Health Canada authorized trial in arthritis patients. She's currently investigating minimally manipulated cell therapies in another arthritis trial. She has several leadership roles, including as co-chair of the scientific MSC committee and as VP-elect for the North American region of the International Society for Cell and Gene Therapy from 2024 to 2026, and is going to be the VP for 2026 to 2028. So take it away, Dr. Viswanathan.
Thanks, Matthew. Happy to be here. And thanks to Stem Cell Network for organizing this webinar. So I'm going to pick up from where Megan and all of your excellent speakers that you've heard about and talk about a clinical trial, which is the minimally manipulated one that you heard about. And I've titled my talk, "If at first you don't succeed, submit another amendment." And I hope you'll see what I mean by the end of this. So the trial is really focused on arthritis, and I just wanted to give a quick primer on the fact that we have more patients with osteoarthritis than we do patients with heart disease, cancer, stroke, and diabetes put together in Canada. And this statistics is true globally as well. And this is actually a growing epidemic because unfortunately we have an aging as well as an obese population. And for a long time, osteoarthritis was sort of thought of as an older person's wear and tear mechanical disease, but now it's understood that it is a low-grade inflammatory disease with a huge metabolic dysregulation component. And really, even though we've had osteoarthritis for thousands of years, interestingly, when you look at kind of old archaeological digs and they find bones, they find that those people had osteoarthritis. We still don't have anything other than kind of management of the disease. So typically sort of various types of pain management and symptom management with some sort of short-term relief injections. And then as patients sort of progress and live with this disease, essentially for decades, they qualify for a joint replacement, which is a prosthetic joint that they get. But there's really no disease-modifying drugs that are available to reverse the progression of this very degenerative disease. And that's not for the lack of trying, right? There's been decades of research and billions of dollars invested into this. But the modality of this disease and the fact that it affects all of these different tissues other than the cartilage has really proven to be very challenging. And many of these trials that have tried to identify single targets and go at them with small molecules or cytokines or other approaches have largely failed in sort of placebo-controlled large trials. So there's this huge unmet clinical need. So it was against this backdrop that we got Health Canada authorization for conducting this fairly large statistically powered clinical trial where we're taking bone marrow from the patient and very minimally processing it and then injecting it back into their knee. And this is a randomized, double-blinded, placebo-controlled clinical trial where we're following patients out to a year, but their clinical endpoint is around 6 months. I want to talk to you a little bit about the rationale for this trial. So as I said, there's a few injections that patients can get: corticosteroids, hyaluronic acid, and sometimes platelet-rich plasma. This provides short-term relief, and the efficacy for most of these is highly questionable. And this is more problematic because many of these patients pay for these treatments out of pocket. So to have these treatments that are available, but that patients have to pay, but there is really very limited efficacy data is, as I said, problematic. What the field really has been lacking, despite tons of clinical kind of data, there has not been very well controlled, rigorous, randomized clinical trials, which can really properly address questions of efficacy. Bone marrow aspirate was offered in Canada until 2019 when it was withdrawn, and now can only be offered under the auspices of a clinical trial, such as the one we're conducting. And the reason for this is because I think it was falsely marketed as a stem cell product. And this is disingenuous given that about 0.01% of your bone marrow contains so-called stem cells. So it's 99.99% non-stem cells. So to call it a stem cell product, I think is problematic as well. Anyway, we believe that bone marrow aspirate actually contains many different cell types, including hematopoietic cells, endothelial cells, and stromal cells, which secrete various factors. And prime among these, we think IL-1 receptor antagonist, primarily made by monocytes and macrophages, are really important and can kind of combat inflammation and provide pain relief and symptom relief. So while these therapies exist, societies like the Canadian Association for Arthroscopic have put out position statements and essentially have emphasized that use of sort of these bone marrow injections cannot be recommended at the time. The research is promising, but really needs more rigorous trials to kind of prove this. And these sentiments are echoed by other societies, including the American Association of Hip and Knee Surgeons, where, again, they emphasize the need for rigorous, well-designed clinical trials to be able to address these gaps in the field. And that's precisely what we're trying to do in our clinical trial, where we've hypothesized that there's going to be a 35% difference in the efficacy rate between patients that receive this bone marrow cellular preparation versus the placebo group in terms of their response rates to something called the numerical pain rating score, or NPRS. And I'll talk about this in a minute. And we hypothesize that really the difference is being driven by baseline immune profile in these patients, particularly their monocyte macrophages, T helper cells, neutrophils, and NK cells. So those are sort of the 4 different panels. And then we have subsets within these that we profile in these patients, both at the circulating level, but also specifically in the joint. And we also think that there is heterogeneity in the preparations of these autologous treatments because everyone's bone marrow is slightly different and has different cellular components, which then results in different soluble factors and potentially different kind of transcriptomic signatures that these preparations carry. And with omics technologies, we can now be able to parse these different types of signatures in the cellular preparations. So this is sort of a very data-heavy trial that we're proposing to not only do this very statistically significantly powered clinical trial, but also to look at why we have these sort of responders and non-responders and base it on sort of biological data that we're collecting and samples that we're biobanking to be able to do this at a very large scale. So what I thought I would present for the talk is sort of 5 lessons that we've learned in the process of launching this clinical trial and further. And some of these items were brought up by previous speakers. So I think I'll be able to illustrate this with example. One of the big things that we had to really decide was kind of the study design, right? And choosing the primary endpoint was very important. So pain was the primary endpoint that we chose because it's clinically relevant and very meaningful, but also one that's highly subjective and variable. And while there are different instruments for measuring pain, again, these are patient-reported outcomes, and we had to choose the instrument that we were going to rely on. And we chose this NPRS scale, which I'm sure most of you are familiar with. This is a 0 to 10, 11-point scale. And the reason we chose this is because it is so familiar and very intuitive for patients to be able to provide answers on. But because it's 11-point scale, it has very limited sort of range in terms of providing us with data. And so we needed to choose sort of a 2-point cutoff or a threshold in improvements that was clinically meaningful. Speaking with clinicians, they also wanted to kind of come up with a response rate. So the rate in which patients in the treatment arm versus patients in the placebo arm responded, and they wanted to see a significant difference in that because that was something that they were used to seeing and was, again, something that would translate more meaningfully into the clinic. When we went to Health Canada, they wanted to see an absolute change in pain scores, not this sort of percent improvement. And they based it on other guidelines. Erin talked about FDA guidelines, but in this case, Health Canada referred us to EMA, European Medicine Agency guidelines that speak to sort of using absolute change in pain scores. So we actually engaged with a number of stakeholders, clinicians, our scientific advisory board, and with Health Canada through a series of these pre-CTA meetings that I'll talk about in a second. And we finally compromised and kept our NPRS as our primary endpoint, but we added a secondary nested endpoint, which was this absolute change in pain. And this had implications in terms of our sample size calculations, which then affects our timelines and costs for running the clinical trial. And we had not one, not two, but three pre-CTA meetings. And you can see that this spanned over a period of several months, because for each of these meetings, you really need to prepare a package and a dossier, because Health Canada is not really subject matter experts on these discussions, right? So you're asking them to weigh in on their opinion, but you need to give them enough background and enough context to come in and weigh in on this. So there's a file or a package that needs to be prepared each time. These meetings are formal, they're minuted. Both parties, Health Canada and you, have to agree to these meeting minutes. So this took us some time, and we took all of these learnings and input before we finally submitted our CTA in March of 2022. Okay. So we got our CTA approval. It's a no objection letter in June of 2022. We immediately had to do an amendment, and I'll talk about amendments. And then we wanted to go get our ethics approval. So in Ontario, we applied for something called Clinical Trials Ontario because it allows us to do approvals across multiple institutions and hospitals, which our trial was. It was a multi-site one, but we still needed several ethics approvals because we also needed our host approvals. Interestingly, the ethics approval took us much longer than Health Canada. It took us between 8 to 9 months to get ethics approval. And when we were really ready to start our trial in 2023, we ran into a huge roadblock. So Megan talked about rapamycin. We ran into supplier issues, and I'll bring this up in a minute. But essentially, our critical supplier backed out of distributing in Canada, and we had to pivot and find a new supplier and a new vendor, but that meant changing our entire process. And so we had to do a comparability study to show that the new product was comparable to the old product, and do a big amendment after we had this comparability data. So that delayed us by a year and a half before we finally were able to recruit our first patient in 2025. We have since screened 258 patients and consented 24 patients to date. So that was sort of one lesson, is that it takes, as Megan mentioned, a lot longer to get your design out, but you also need to get multiple stakeholders into play get all of your design considerations. The second lesson that we learned the hard way was supply chain issues. And as I said, our whole product and process was based on this product from Terumo BCT, where you take the bone marrow and essentially you're interested in the buffy coat, and you centrifuge it to get this concentrate, which contains all of these different cell types, but also the IL receptor and PDGF growth factors that we were interested in. Terumo BCT sold this part of their business to another vendor, and the other vendor decided that Canada was too small a market and not worth going through the regulatory hassle, and so they pulled out of distributing in Canada. So we needed to pivot, and we moved to this servos device called Marrow Solutions, which was actually a very different procedure of aspirating the bone marrow from the patient's iliac crest, and comes with these multiple pinholes, which aspirates very small volumes of bone marrow. And essentially what this means is that there is no centrifugation required. So it's already in a concentrated form because it pulls out small samples. And so we had two very different procedures and had to do a prospective comparability study to show that the two procedures were equivalent. And so this took us about a year to generate this data, where we had 5 different donors that consented to have their bone marrow donated, but not used for the trial, but just for research purposes. And we basically did a split comparison between kind of the servos and the Terumo devices, and had to come up with metrics because not a lot of it's established in the field using total nucleated cell counts and CD34 cell counts. And you can see that there's a lot of variability between the donors. And essentially, we had to argue that the variability between the donors was greater than the variability between the two different procedures. And this is what we ended up doing in our comparability study. We had a lot more metrics than this, but I'm just showing you a snapshot of what we had to essentially deal with. So this took us about a year and a half to figure out. So like Megan, I want to emphasize that when you have key reagents, it's really important to do your qualification. It's more expensive and will take you more time to do this. But in the end, this might be time saving because we had to do it at the back end. And that was a lot more time and money than if you had to do it upfront. Okay. Our trial is rigorously conducted, so it's a double-blinded study. So neither the clinician nor the patient knows what they're receiving. But the reason for this is because in musculoskeletal trials, and especially those that are procedure-based, there is a huge placebo effect. So there's kind of like a drug which is described as a placebo, or when it's given as an OA treatment, or when an OA treatment is given as a placebo. There are all of these effects, and really to get at the total effect, you need to be able to subtract this placebo effect. So I'm sensitive to time, so I'm going to zip through this a little bit. But essentially what we needed to realize very quickly is that we needed 2 teams, right? A blinded team, which was the processing team, and then the clinical team, which was really, the clinical team, which was blinded, and the processing team that was unblinded. But this meant really configuring our workspace in a busy hospital fracture clinic and having 2 dedicated examination rooms to be able to control this workflow and sample flow from one room to the other, and ensuring safeguards, for example, having the syringes blinded and properly labeled. And for example, having the syringes primed, so the clinician wouldn't be priming the syringe and seeing the little squirt of blood in the bone marrow versus, just saline coming out in the saline syringe, which would kind of give it away to them. Now, there's still tactile differences for the clinician that's injecting it, which we can't fully overcome. So it's not 100% double-blinded, but the patient is fully blinded to this. But it also meant that all of our SOPs and that we developed really needed to have this blinding in place. And we had to do multiple dry runs to make sure that all of our process flows were done with regard to this blinding in place. So the practical implementation of this blind was really important. Lesson 4 is really the recruitment challenges that we face. So even though we took us years to kind of get us started, we were really happy. We thought we'd be very rigorous in terms of getting recruitment, and we certainly had a lot of patients interested, but you can see that the recruitment rate is really 1 in 10. And there's a lot of reasons for this, primarily the inclusion-exclusion criteria, which is tight. And also because many of the patients that we see in the hospitals are not really ideal candidates. These are patients that want surgery, they want pain relief immediately, they don't want to be randomized and have a 50% shot at what they're getting, and sometimes they're on contraindications. So what this taught us is that we really needed to go out into the community and build a network of community clinics and partners that could refer to us and use all of the available methodologies, including newsletters and other means to kind of promote and get visibility for our trial. The last lesson is that this is a living document, right? So just because you have your CTA submitted and done, you're not out of the woods, right? So we've done several since then, including changing kind of the vendor, but also relaxing our inclusion-exclusion criteria when we ran into inclusion recruitment challenges, right? So we realized that this is very much a living document and we need to kind of be iterative about this. So I'll summarize here because I know we're running out of time, but essentially what we're doing is demonstrating and developing robust data, which we can use to either show or disprove efficacy, but our data we think will be reliable because we're taking all of these measures to do this in an unbiased manner, and also because we're looking at all of these other biomarkers to help us understand the heterogeneity in responsiveness. But many of these, even though we went and thought we were very considerate in our design of the clinical trial and had many stakeholders weigh in, many of these design considerations are often not practicable and implementable. And so you really need to stay nimble, and that requires adaptation and iteration. This is for the trainees, just so you know that it's a very multidisciplinary team. So we have regulatory support, we have the processing team, which is more on the GMP side. We have the clinical team, we have our vendors, and then of course we have kind of the research and the analytical side of things. And none of this would have been possible without Foundation and Stem Cell Network support. So I'll acknowledge that and stop sharing so we can maybe have one or two questions. Matthew, over to you.
Yeah, thanks a lot. So Ellie indicated that, we're going to keep the Q&A open till about 2:40, but I think if people can't stay, feel free to leave. The other speakers have been working really hard, I can see, to answer as many questions as they can. So if you asked a question, it might be answered in the Q&A. Have a look for that. And I'll pick out a few so we can have a question or two before we close. And I'll be selective about that. So one that jumped out to me, it's a brand new question, but speaks to me maybe. As a trainee, if we're working on a translational project that teaches us how to move basic science into the clinic, learning about preparing data for CTA, pre-CTA, Q&A, what career paths does that sort of qualify us for? And I guess maybe to add on to that, how can trainees get more exposure to this kind of all the things that we've been hearing about today in their academic training? Open to anyone, I think.
I can take a stab to start, maybe if so I guess speaking to the part of your question about getting more exposure, I think maybe one of the ways I would say is to just engage with networks like Stem Cell Network and BioCanRx that are kind of tapped in to some of the programs, the training programs, the mentorship programs that are available, because there are things that are available out there. And if you engage with networks, we can help point you to those, and we're very happy to if you want to connect with us after the call. We're involved in something called CanPrime, which we support for hands-on biomanufacturing experience. And there's Castle, there's Caddy, there's like all sorts of things. Just to point out a few, there's a science to business mentorship program. So lots of ways to kind of get involved.
And to add to that, at NRC, we are also, involved in training students. So we have training opportunities either as summer students or WAPs or trainees that come for graduate students that, we do translational research here. So students are welcome to apply and come and get trained.
Maybe I'll just comment that something that I didn't realize before I got into this is how many different job opportunities there are in this translational medicine space. You saw that last slide that Somya had, like all the different people, teams that were involved to get this large trial going. And so, all the way from maybe doing the GLP toxicology studies, like hands on stuff, doing the actual manufacturing in the GMP facility, doing the quality management, doing the clinical trial, writing the documents, the regulatory support. The list kind of just goes on and on. And you can do those jobs with some jobs with only a bachelor's, some with a master's, some require a PhD. So there's just so many different opportunities you can develop your career in this area.
One more question that I'd really like to hear your thoughts on. For regenerative medicine therapies, what is everyone's perspective on the most fatal clinical development issue that you've experienced? And maybe on the flip side, uh, for people doing this kind of research, what should we be doing to set ourselves up for success and try to avoid those problems with translation down the road?
I mean, I think it's funding is really important. And to Megan's point, you always always underestimate how much it costs. It costs like um, more than you can usually budget, and so you typically run out of funding or resources or support for this. And if you have delays, that's very costly because it costs you time and money. And again, that kind of speaks to this whole aspect. And I think, the funding environment is such that you really need to hit kind of success to ensure continued funding. But a lot of what you learn, like any experiment that you do in the lab, is iterative, right? So unfortunately, a lot of these, if not most of these clinical trials fail. But that doesn't mean that you don't learn from them, right? But it's very difficult to kind of get the second trial funded if the first one doesn't reach its endpoint. And this is particularly true when you start ramping up and kind of getting into later phase clinical trials. So, what we're trying to do is understand, like our trial is set up so that, yes, it meets its endpoint, but it's still a success whether it's efficacious or not, because I think it shows something and we learn something from it. And by kind of incorporating things like biomarkers, that helps us understand, okay, if there is heterogeneity, why is that heterogeneity happening and how to understand it, then we can kind of build off of that and move to later phase clinical trials. But the field itself is not really set up for an iterative clinical approach. And I think that's really where we are in terms of how we're learning about these advanced therapies and how they work in humans, and which is very different from however well they're modeled in the labs.
Mhm. Megan, you have something to
Yeah, I
add?
think just to expand on that, you need to have that end clinical application really solid. I think a lot of times people start off with their cool, whatever thing they discovered, but they haven't really thought through, okay, if I was going to use this in a patient, would the healthcare system pay for it? That's like ultimately the question you have to ask. And so, um, especially in the Canadian context, are you going for the Canadian context? Are you going for the American context? So something that I haven't started to do yet, but I think we really need to start doing more is working with health economists to make that long-term vision clear from the very beginning, because there's kind of no point doing a phase one trial if there's never going to be a financially feasible clinical application. So that's definitely something I've learned.
Erin, you have
Yeah,
final thoughts for us? I think
yes,
we'll have to close.
okay, final thought. No, I just wanted to echo the funding piece. I think in Canada, we're really great at funding the discovery research, but we maybe don't have as much of a kind of coordinated national strategy to fund that translational piece. Obviously, we fund that kind of work at BioCanRx, and I think if Canada could do this collectively to really get the supports and the funding to people who are trying to translate it would also be really helpful.
Okay, well, thanks to all the speakers. I'm going to pass it back to Ellie and Julie to close us off, but thanks for having me for moderating.
Guess just a quick final thank you from me to all the speakers and for your extra time today. You covered so much ground in your talk, so thank you for putting all of that content together and for the additional insight and the Q&A. And just a final note to the attendees, if you want to get in touch with BioCanRx, follow us on social media, join our newsletter, reach out, and hope to see you at future events.
Yes, thank you. From Stem Cell Network, I wanted to say a final thank you to all the speakers, to Matthew for expertly moderating the session, and to BioCanRx for being a wonderful co-host. And a final plug for the post-event survey as well. After the session closes, we'd really appreciate if you take a couple minutes just to complete that survey because your feedback is essential to help us plan future events. And make sure you check out all of our other opportunities through the year at stemcellnetwork.ca. So with that, we'll close. One final thank you and have a great day.